everybody, and welcome to April's edition of the Global Nexus Conversations on Acute Infectious Disease Threats. My name is Mike D'Agostino, and I am a PhD candidate in Dr. Matthew Miller's lab, and I will be the moderator for this afternoon's session. To get things started, I would like to recognize that here at McMaster, we are located on the traditional territory shared between the Haudenosaunee Confederacy and the Anishinaabe Nations, which was acknowledged in the dish with one spoon wampum belt. That wampum uses the symbolism of a dish to represent the territory and one spoon to represent that the people are to share the resources of the land and to only take what they need. Today, we are very fortunate to have 4 panelists, most of whom I've been lucky enough to have the opportunity to work with in the past. Importantly, our panelists' extensive expertise spans a range of topics, including the emergence of new infectious diseases from animal reservoirs, the importance of early detection and surveillance, and vaccine development and distribution, and the impact of infectious diseases on global health with a focus with a One Health perspective. Our first panelist is Dr. Arinjay Banerjee, who is a principal scientist at the Vaccine and Infectious Disease Organization and adjunct faculty member at the Universities of Saskatchewan, Waterloo, Toronto, and British Columbia. Dr. Banerjee is the co-lead for One Health at the University of Saskatchewan, and research within his laboratory focuses on 3 main themes that are inspired by the One Health ideology. The first being virus-host interactions in wildlife reservoir species, such as bats. 2, virus-host interactions in spillover species, such as humans. And 3, viral vaccine development. Secondly, I'd like to introduce Dr. Matthew Miller, who's an associate professor in the Department of Biochemistry and Biomedical Sciences here at McMaster University, as well as the scientific director for the Michael G. DeGroote Institute for Infectious Disease Research, or IADR. Dr. Miller is also a core member of McMaster's Global Nexus for Pandemic and Biological Threats. And in his laboratory, we study diverse aspects of virus-host interactions, including the development of broadly protective vaccines against respiratory viruses, such as influenza virus and coronaviruses. We study the biology of broadly neutralizing antibodies and the importance of infection and inflammation on neurodegenerative diseases, especially ALS. I would now like to introduce Dr. Zvonimir Poljak, who is an associate professor in swine health management and epidemiology in the Department of Population Medicine at the University of Guelph. His current research broadly covers epidemiology of respiratory infections, improvements in the surveillance of animal diseases, and planning mitigation strategies for the disruption of food supply chains. Dr. Poljak has been involved in research related to several animal health emergencies in Canada and abroad, including different subtypes of influenza A viruses and coronaviruses in swine in Canada, and emerging vector-borne diseases of ruminants in Croatia. He is also a research program director for the Ontario Agri-Food Innovation Alliance in the area of animal health and production, and is currently acting Associate Dean, Research and Graduate Studies at the Ontario Veterinary College. And last but certainly not least, we have Dr. Fiona Smaill, who is a Professor Emerita in the Department of Pathology and Molecular Medicine here at McMaster. Following an active career as an academic infectious disease physician and microbiologist, her interest now is supporting clinical trials of mucosal vaccines for infectious diseases such as tuberculosis, and she is presently leading McMaster's phase 1 and 2 studies of an inhaled COVID-19 vaccine. As we all know, infectious disease remains a major global health concern, with the COVID-19 pandemic highlighting the need for strong public health preparedness and a harmonized response between sectors. This session aims to shed light on the current state of infectious disease threats, as well as the strategies and innovations being employed to mitigate their impact. We hope that this conversation will provide a valuable opportunity for the attendees to learn from and engage with our panelists, as well as to gain insight into the latest developments and challenges in infectious disease management. We invite all attendees to please post any questions for the panelists in the Q&A box. Alternatively, you can feel free to raise your hand and I can then call upon you to ask your own question if you're comfortable. To get things started, our first question today is what type of virus or what animal reservoirs are the next potential outbreaks going to be coming from? And what kind of work is being done in this area to help us prepare? Dr. Banerjee, if you would like to please get us started.
Thank you, Michael. And well, my laboratory studies bats, so I'll be biased and give a biased response that perhaps wildlife is going to be the source for the next virus. Having said that, I think there needs to be more investment into surveillance as well. And oftentimes I use this analogy, you can't fight an enemy you can't see. So we need to be able to identify viruses that do exist. And once we've identified those viruses, we can come up with strategies, perhaps vaccines and frontline therapeutics to tackle those pathogens, but we can't go in blind. So I've had a very deep appreciation of colleagues who do field disease ecology and colleagues that do epidemiology in the field as well. Because we are a laboratory-based research lab, and we are very good at studying viruses and how they interact with their wildlife hosts. But we can only do that once we identify those reservoir species and identify those pathogens with zoonotic potential. So I will continue to push for more support for studies looking into wildlife zoonotic sources of pathogens. And we are aware that there are livestock and other animal species as well that are very important. But that's an angle that I'll probably take. Thanks.
All right, thank you. Dr. Miller, if you'd like to go next.
Sure. Thanks very much, Mike. Thanks for the introduction, and thanks to everyone who tuned in today. It's a pleasure to be here with these other phenomenal colleagues from across the country. I think when thinking about what the next pandemic will be, what I've learned over the course of my career is to be extremely humble. Prior to the last 3 years, my expertise was really in pandemic influenza, and recalling back to 2009, I think there was almost no one who would have guessed that the 2009 swine flu pandemic would be caused by an H1N1 influenza virus, right? Because this was a subtype that was already circulating seasonally. And so very, very few people imagined that it had pandemic potential. Similar to the situation we're in now, everybody was very concerned about H5N1 viruses. There had been a lot of focus on highly pathogenic avian H5N1 in the early 2000s as a result of some really serious epidemics, primarily in Asia and Southeast Asia. And obviously, right now, we're experiencing a really unprecedented global surge in highly pathogenic avian H5N1 in wildlife across the world. It's currently resulting in a tremendous amount of deaths of wildlife, coastal animals particularly, but also a lot of scavenger animals have become infected, and many people will be aware that in Ontario, there was a recent report of a domestic dog that was infected in Oshawa, which is very close, obviously, to many of you who are tuning in today. So I think that Arinjay is right on. I think, you know, the human-animal interface is where zoonosis tends to occur, right? And it is these zoonotic pathogens that tend to have the most pandemic potential. When we look forward to 2019, just like in 2009, I don't think any of us were expecting a coronavirus to cause a pandemic. We had experience and very unique experience in Canada with SARS coronavirus 1. And thankfully, we mitigated disaster back in the early 2000s when that virus arrived in Canada. And I think the relatedness of SARS-CoV-1 to SARS-CoV-2 initially led many experts to be very hopeful that we could similarly limit the spread and the impact of SARS-CoV-2. And even though those viruses share tremendous similarities in their biology, there is obviously tremendous differences in their eventual impact. And then I guess the last point I want to touch on is an issue that may not actually qualify as a pandemic in the strictest terms, but I think is something that we in the Northern Hemisphere will become increasingly burdened by as a result of climate change, is vector-borne pathogens that have been a long-standing and persistent problem, especially in equatorial regions in the global south. But as those insect vectors like mosquitoes, continue to move north as a result of climate change, we are going to be experiencing much more threat from pathogens like dengue, malaria, chikungunya, all of these things that have been sort of a burning problem in other areas of the world for a long time. And we're certainly not ready for that. And so I think it's a really critical area to focus on. And like Arinjay, I think all of this requires truly multidisciplinary and multi-sectoral approaches, because of course there's a lot we can do scientifically and medically to prepare, but there are all kinds of other considerations like implementation, outreach to marginalized communities that require people with expertise that's different from medicine and biomedicine, but is highly complementary.
All right, thank you, Dr. Miller. Dr. Poljak, you can please take the floor.
Sure. Well, first of all, thank you for the invitation. It's really great to be here and to have this opportunity to engage with individuals who have expertise in this emerging infections. I think I would mimic everything that was said so far, and maybe what I would say is, if you're thinking about predicting, of course we have to be humble, but the way that I would divide that is in kind of a short, what do we expect and what is the immediate threat in terms of short term, what is kind of a medium term concern, and then on a long-term basis, what can we expect given that we have global change in climate and so on. So, from a short-term perspective, I think the obvious threat is highly pathogenic H5N1. It's threat to our agricultural species. It's also threat to wild mammals. And I think it has some tendencies that are concerning because of, at least at this point in time, molecular evidence that it could become more transmissible to mammals. Maybe not between mammals, but certainly to mammals. I think what we are missing at this point in time is some sort of peer review evidence about the transmission between different mammalian species. And there are, I think, a couple of mammalian species that we really have to be concerned about. Not concerned maybe immediately, but we should be closely monitored. So we need to monitor these phenotypes. And one of them is definitely ferrets, because they represent tomorrow for humans. But also we have to be interested in a couple of livestock species, because they live in high density populations. And if these viruses gain potential to be highly transmissible in these intensely raised populations, that obviously raises concerns for these animals, but also for humans. And I think we could potentially be talking about completely different scenarios where the bridging populations, or the people who are taking care of these animals, would be exposed to higher level of risk that we ever seen in the past. So we should be, on a short-term basis, we should be really, I think, focusing on H5N1 and developments in that front. I think that on a medium-term basis, coronaviruses and influenza viruses in general should be monitored very closely. And I will give you my experience with coronaviruses from swine. Everything that we've seen in human populations, we have seen over the last 3 years, we've seen pretty much the same things and even more complex in swine populations over the last 3 decades. And just to give you some examples, we have coronaviruses that are mostly spread respiratory, but could affect and cause clinical signs and GI signs and potentially respiratory side, all by the same virus. We could also have strains of very related viruses that are showing different transmissibility and different pathogenicity in the host. We could also see viruses that recombined. And I think we've seen and we haven't seen that in human populations, but we've seen viruses that emerged as GI, gastrointestinal viruses, and then they mutated into a respiratory form. So, you know, on a medium-term basis, definitely coronaviruses would be something that I would be watching very closely. I wouldn't, on a medium-term basis, also ignore influenza viruses, because even outside of H5N1 situation, we have very active dynamics of influenza introductions and sometimes apparent eliminations. For example, relatively recent introduction of H3N2 virus into canine populations in Asia that then spread into North America and now seems to be a dominating virus. Again, unexpected situation that nobody could foresee. I will also finish the last thing, and this is on a long-term basis, we should not be ignoring other viruses either. And again, giving you one example from swine populations, porcine circovirus type 1. We knew about this virus for decades. It was really a contaminant of different cell lines. And then at one point in time, it emerged, another type of circovirus emerged and caused global panzootic, causing very severe clinical signs, is under control due to development of really, really good vaccines. But this is a DNA virus, and nobody really could foresee that something like that would be coming. So the bottom line is we should be monitoring pretty much for everything on a long-term basis and developing different surveillance and monitoring tools, diagnostic tools to allow us for that.
That's a great point. I think that's something that we can look to. The international efforts that go towards influenza surveillance are, you know, one of our top tools and one of our main early warning systems that we have in case of a new influenza outbreak. And we need to look into potentially setting up similar systems for other virus types, such as coronaviruses that do pose a similar threat. And lastly, Dr. Smaill, if you'd like to.
I think my colleagues have really stressed the importance of really the next pandemic coming because of the close proximity between animals and humans. And that's really been our experience with the recent experience with transmission of infectious diseases. I think SARS-1, SARS-2, I also am aware of MERS, we've got influenza, we've heard about the pigs. I also want to just pick up on the point that Matthew made about vector, and it's actually not just the Northern Hemisphere, because Japanese encephalitis, which is a pig virus, traditionally in Asia, has been seen in the suburbs of Sydney in Australia. So climate change, and I think that's the cause for it, is a vital part of what we have to be on the lookout for. Because I'm an infectious disease physician, I take a slightly different perspective, because I'm looking to see what do people do to make a virus that has perhaps transmitted from animals to a human host in, say, the market in Wuhan or in the camps in West Africa. And there are also, of course, sexually transmitted infections. And the first pandemic that I was involved in was, of course, HIV. And in those early days, there was really very little understanding of how it was transmitted and what the implications of it was. So yes, respiratory viruses are predominant when we're thinking about the type of infections we're going to see, but we shouldn't forget the behavioral characteristics of people. We travel the world so quickly. We also, and we've seen the transmission of sexually transmitted infections. More recently, we had Mpox, and the questions related to how was that going to be transmitted. So when these viruses sort of jump into humans, it then takes really that next stage is a behavioral component, an epidemiological component, a social component that then really gives rise to what we see now as pandemic. So I just want to take it to that next stage, and I'll probably stop there, and I'm sure there's questions that will follow to the group of us.
Nope, those are all very, very good points, Dr. Smaill, and you know, that's one of the benefits of having people from both sides of the bench and the bedside, is that you're there to offer that perspective, and that really offers some very different insight that I'm sure not a lot of us are exposed to all the time. So we have 2 questions in the chat right now. The first of which is, how dangerous are co-infections of influenza and SARS-CoV-2 variants? Dr. Miller, I'm assuming you might want to jump in on that one.
Yeah, I mean, it's a good question. And to be frank, I'm not sure we know with the degree of certainty that we would ideally like at this point. You know, really, we've only experienced any appreciable co-circulation of seasonal influenza virus with SARS-CoV-2 over the past, year or so, after the sort of relaxing of public health measures facilitated the recirculation of seasonal influenza. We haven't seen, I don't think, epidemiologically hugely worrying signals yet, but there's considerable uncertainty. There are obviously examples of situations where infection with one pathogen creates some degree of protection, often innate immune mediated, against another pathogen. But there are also situations where a particular pathogen can increase susceptibility. There have been some reductionist studies in vitro that have looked at co-infections of influenza viruses and coronaviruses. I'm aware of one specific one that comes to mind that was completed by Guy Boivin's lab at University of Laval quite recently, and actually found a very interesting temporal relationship when looking at which virus came first, flu or coronavirus. But yeah, certainly from a clinical or epidemiological perspective, I'm not sure we fully understand that yet. And it's also a very nuanced question because it depends inherently on what demographic we're talking about, right? So with influenza virus, we see very young children, usually between birth and 4 years of age, are a particularly high-risk population. And then we start to see risk increase quite steeply above age 65 and especially above age 80. That elder population who's at high risk is similar to the high-risk population we see with SARS-CoV-2, but young children are discrepant, right? We don't see that elevated risk to the same extent with very young children in the context of coronavirus. So I think it's a really important question, but I wouldn't bet my house one way or the other yet. I think it hasn't been sufficiently studied in humans at epidemiological scales.
All right, thank you, Dr. Miller. Dr. Banerjee, Poljak, Smaill, is there anything that you feel Dr. Miller might have missed on that? Arinjay, I see you're unmuting.
No, I think I fully agree with Dr. Miller. He's covered it all.
There was actually a bit of a, yeah, sorry, go ahead.
I was just going to say, it was interesting, prior to COVID, there was actually some belief that one respiratory virus might protect against the other. I don't think it ever panned out. And as you probably recall, there was a lot of concern that we were going to have the dual influenza and SARS decimating the population. It never really eventuated. The uncertainty is whether it relates to just how we were avoiding transmission because of the steps that were in place to prevent SARS, whether that COVID infection, whether that was beneficial for influenza. So I think the jury is still out on it. But my general sense is that it's not something we need to panic about at the moment.
All right, thank you, Dr. Smaill. Dr. Poljak, is there anything you might need to add on to that, or are you happy with your colleagues' answers?
Of course, I don't think that I have much to add.
Mike, if I can add something, very early on during the pandemic, we worked with our colleague, Dr. Miller, and we hypothesized that MERS-CoV and SARS-CoV-2 could potentially recombine. And at the time, that was a huge risk that we were worried about, but we haven't seen that yet. So I think even within coronaviruses, there are things we don't fully understand. And it may not be as simple as co-infection or co-infection causing recombination of two beta coronaviruses.
And there was that great concern that with the World Cup and everybody with COVID infection coming to countries where there was MERS, that this would lead to an explosive outbreak. We never saw that. So I think what was proven epidemiologically was what perhaps was being thought, or else there's not a lot of MERS circulating, which is probably true as well.
Yeah, Arinjay, I actually remember you sending me a draft of that prospectus paper that you were writing very early on, talking about the dangers of recombination for coronaviruses and how it was very overlooked by everyone. And we're seeing that now with the majority of the dominating SARS-CoV-2 lineages that are currently circulating being these recombination variants. So we have another question now, which is, how can communication be improved during these acute disease outbreaks? And what is the role of us as scientists in pandemics or outbreak communication along with combating misinformation? I know, Arinjay, you have a quite extensive Twitter following and you're very big on science communication. So maybe you want to take the floor to begin this session.
Oh, I've got colleagues who are virology superstars, but one thing that I appreciate now, Michael, is how collaborative we can be and how collaboratively we can do science that's potentially more translational than what we would have done independently or individually. So that's, we probably lost Michael. So that's something I've come to appreciate deeply. And I'm a very collaborative scientist myself, and I'm continue to build that and hopefully translate that in my trainees as well, to have that ideology of being collaborative and working together as part of a team. Science communication, I don't know. It does come with, it's a double-edged sword. You try to do your best job at it, but a lot of people may come at you for doing it. So it really depends how thick of a skin I or my colleagues can develop. If you're okay with being heckled and harassed with the other part of the community that may not necessarily agree with your science or with our science, then that's fine. But I've taken a step back because it just wasn't worth my mental health. So I don't actively do as much science communication that I perhaps would have done. But that's just my opinion. Let's see what my colleagues have to add to it.
All right. Thank you, Dr. Banerjee. Dr. Poljak, you seem to have a lot of communication is going to be necessary in your line of work, dealing with talking to the agricultural sort of regulatory bodies of the government as well as food supply chains. So how would what's your view on communication during these zoonotic outbreaks?
I think I have multiple thoughts. One of them is that when things are developing with such speed and velocity, and we don't know all the facts, this is a difficult position to communicate anything. So I think any person who is doing communication should learn, and I'm not saying that I've learned it, but should be aware of how to communicate this uncertainty to different types of, different groups in the society. general public versus agricultural groups or government officials. They're all our audience, and I think we always need to learn how to, especially with public, how do we communicate this uncertainty? And the fact that we just don't know certain things when things are developing and changing on a day-to-day basis. And that, I think, leads me to the other thing. How do we communicate, these things that could be changing, right? We know something today, we know more tomorrow, and what we know tomorrow may be a little bit different from what we knew yesterday. So that, I think, is a big challenge. The other one is perhaps that communication, in any kind of emergency and crisis, it's almost a part of this incident command system structure where you have people who are part of the communication, and everything, in order for emergencies to be managed appropriately, everything should be somehow managed within this ICS system. So if you have, too many noises because of these uncertainties that will happen, that could potentially lead to some miscommunication. My other thought is that some of what you touched on was misinformation. And this misinformation is so damaging. And some of that misinformation is deliberate. And some of that misinformation is not. It's just part of human nature. And I think I'm not a communication specialist, but we should have people, and they're already studying that, but we should increase our efforts in order to understand the origins of this miscommunication. And potentially, there is maybe area of natural language processing and ethical use of artificial intelligence in order to combat that. But we need to understand the reasons and the origins of this misinformation.
Yeah, thank you. All very, very valid points there. The source of the misinformation, particularly a big one, and that's one that's very detrimental. And it is a bit of a juggling act, because you're all experts here in your fields, but as you're working with the information that's being updated so rapidly that it's almost impossible to keep up with, it's sort of hard to, have to go back and explain how maybe something you said one day is wrong based on the new evidence that has emerged since, and you have to sort of backtrack and, simultaneously stand by what you said and say that it was supported by evidence at the time, but also be flexible enough to admit that, okay, maybe some things have changed and, new light has brought some other evidence forth. All right, Dr. Miller, I see you've unmuted there, so if you want to go next.
Yeah, no, I think Dr. Poljak is right on. I think that one of the most important things we can do to enhance the effectiveness of our communication in the future is to do a much better job teaching science literacy at the earliest possible stage, right? Because the reality is that most people in the general population don't have university level science training. And so we need people, at the level of, elementary school and high school to understand that science is a discipline that evolves. And I think historically we've taught, science as just these blocks of discrete, unchanging facts. And I think that has done us a disservice in situations where we're having to adapt and do science on the fly, which is obviously sort of not the normal way that science is done. I also think that although I completely agree that scientists have a responsibility to help communicate science, I also think we need to be, again, modest and humble and understand that we're not always the best people to do that communication. And that can be for multiple reasons. One can be because we are not always trusted members of every group that we need to communicate with. And so working with trusted members of communities, especially communities that may be, historically marginalized or may have been, historically exploited, for example, by government or the medical establishment, that's really important. And again, there are people whose livelihoods and expertise is squarely in the realm of communication. And so I think we need to leverage that expertise. I also think we have to be really important about understanding the limits and depth of our own expertise. And that's something that I've seen be a real problem during the pandemic, because again, as Dr. Poljak mentioned, there are two types of misinformation. There's the intentional misinformation, but then there's accidental misinformation. And accidental misinformation can be especially damaging when it comes from someone who appears to be credible as a result of their credentials, but may not fully understand an issue. And so I think that is really important as well. And then for me, the most insidious or difficult to address reality of science communication in our current, highly interconnected world is the problem of social media, and the fact that there's no way to, at least now, there's no really good ways to effectively curate and control what is reliable information. And so that loops back to my earlier point about science literacy. We need to teach people to understand that science evolves, and we also need to teach people how to find credible information and how to understand the realities of scientific consensus, so that they aren't confused when they hear dissenting voices that represent a minority or an extreme view. And then the last comment I'll make is that we've seen this pandemic unfold in a world that is increasingly impacted by populist politics, which has the downstream effect of polarizing the population along political lines. And those political polarizations have transcended, I think, in the current pandemic into the medical realm and the scientific realm. And so I think it's extraordinarily important that, we communicate with people with empathy, that we're willing to talk to people who have dissenting views, and that we do so in a respectful way. Because what has happened, I think, and it's really unfortunate, is that there has been a lot of encampments on both ends of the spectrum, and those people will never have the trust or basis to talk to one another, and either respect each other's positions or be in a position to have their mind changed. I think if you're not willing for your mind to be changed, that is always a big problem in science and a big problem in society. And we can't have our minds changed unless we're willing to talk to people and listen to people who have dissenting views.
And just perhaps a couple of final comments. Trust isn't developed overnight. It often takes decades to build up trust with your colleagues, with the community, and particularly policymakers. And I think one of our responsibilities as scientists is to ensure that policymakers get clear information on which they are able to make clear decisions. I think a lot of the challenges that we often see relate to information that seems to be conflicting, coming from different sources. And one of our challenges, I think, as scientists, is to ensure that the policymakers, who really are the ones who are out there front and center of managing many of these pandemic associated events, have the best information, and we communicate them to them in the clearest way. And as I mentioned, it doesn't happen overnight. So ensuring that over our careers, really, we have established that trust with the people who will be making decisions, I think, is a vital part of our communication.
All great points there. So thank you all for your insight. The next question in the chat is, how can diagnostic technologies help for pandemic prevention when it comes to diseases that originate in animals? And should we then actively be surveilling animals, whether they be domesticated or out in the wild? I guess, Dr. Poljak, we'll begin with you for this round.
Sure. I think there are two aspects to this, in my opinion. One of them is we can surveil for things that we know could be a potential problem, and we are doing that in one way or another. But I think what's really important is to have not only the local surveillance, but also to have global surveillance. So I think we are all interconnected, and right now, at least for influenza, there are really good databases. They're still probably sampled in a biased way, so that certain regions are not really represented appropriately. And that's something that should be worked on. And then there are other aspects of pathogens that we don't know that could potentially spill over into humans. And for this, I see, although I'm not a diagnostician, but I see a great promise of some of these new technologies that can sample for a very cost-effective way to see what's actually going on. And I think I would add to this one additional point, and this is with this tremendous amount of data. I think this is one additional point where we can start looking into the use of artificial intelligence and machine learning methods to kind of monitor in real time, because these are literally tens of millions of data points to identify and do ongoing risk assessment. So local surveillance in Canada is great for animals. Globally, I think there's a lot to do. There are new technologies on the sequencing side. I think we have to think about the ways to incorporate risk assessment and artificial intelligence into this risk assessment.
No, that's a very valid point. I mean, it's been shown time and time again now. I feel like every day we're getting more updates on the uses of artificial intelligence. And I think that the sooner we can find ways to incorporate this into our day-to-day lives and our research, probably the better. Dr. Banerjee, if you want to go next, please.
Thank you, Michael. I fully agree with my colleague, Dr. Poljak. But, surveillance is critical, because imagine if you didn't have a database, we had an outbreak of SARS-2, it wouldn't take you days, it would take you months to figure out what that viral family is if that database didn't exist. And surveillance is key to having that database. But how do you incentivize surveillance? Who pays for it? If you look at a country like India, where I'm from, there are bigger problems. There's cholera, there's dengue, there's malaria. Why would the government pay for surveilling the animal populations? And let's say somebody funds it. What are the ramifications of finding a disease that may have trade implications for that country, right? So why would that state government or the federal government allow you to surveil those animal populations? So these are questions that are very tough. I don't understand political science or bilateral ties enough to figure out who's going to pay for it and what are the implications of surveilling animals in countries outside of Canada. Just something to think about, I guess.
Very good point there. And I think that also speaks to our previous comment on communicating. And you're humble enough there to know that you don't quite know all the ins and outs of international relations and policy. So good on you there for recognizing where your expertise comes to an end. Dr. Smaill, if you'd like to go next.
Once we recognize that there is the potential for a pandemic, it's of course very important to have diagnostic testing abilities within countries for people. And we were left scrambling, trying to really ramp up diagnostic testing, particularly in Canada, where our public health labs were really overstretched to pick up what these challenges were. And that's in Canada, where we are well enough resourced. In other countries, the ability to do the diagnostic testing that you need to know you've got a pandemic, and then, of course, to monitor is the other component of this. So I agree that the animal surveillance is vital, but it should be part of a continuum where we've got the resources to be able to really diagnose, with the assumption that there will be therapies, be it vaccines, monoclonals, or antivirals available. But to be able to control the pandemic, you have to be able to diagnose the cases. And that requires a laboratory infrastructure that has to be well maintained into pandemic, if that's the right term.
All right, thank you. And Dr. Miller, if you'd like to finish this off.
Yeah, I don't have a lot to add. My colleagues, I think, have said it all. I would just dovetail from Dr. Smaill's excellent point by saying that if we were wise enough to invest early in generating diagnostic tools for wildlife surveillance, it would massively ease the repurposing of those tools for later human diagnostics, right? If you have a lateral flow test that works for, coronavirus sampling in bats, it's not as hard as starting de novo to generate or adapt that test for subsequent use in humans. And the testing is important, as Dr. Smaill noted, for two reasons. When you do get to the human stage, one is it's critical for surveillance in humans and understanding epidemiology, but later it can become clinically important for decision making, especially because there are only so many symptoms a human can have. And, we have very narrow spectrum antivirals overall. And so, for example, differentiating influenza from a coronavirus infection in certain cases is very clinically meaningful, right? Because it could inform, a course of therapy.
If I can just add one additional point about animal surveillance that definitely is not the case for human populations. And this is the fact that the moment that you detect something that is considered highly contagious, that moment, certain countries could experience trade restrictions. And it took years and decades to build a surveillance system, going again back to Dr. Smaiill's point about trust. For the government organizations, veterinary organizations, and animal health experts to start reporting centrally that something serious is going on in the country.
Thank you, Dr. Poljak. Very, very valid point to add on to the end there. Thank you. The next question that we have in the chat is, how dangerous are gain-of-function experiments, and should they be banned, or what would the regulatory oversight of these sort of experiments be restricted to? Matt, I see you smiling there, so if you want to go ahead and get started.
Yeah, sure. I mean, I'm smiling because this debate has come full circle. When I was a postdoctoral fellow in New York, the real intense recent debates about gain-of-function research sort of hit a boiling point, and it was as a result of some studies related to transmissibility of H5N1 influenza virus that were conducted in the US and the Netherlands. Gain-of-function research is a very and the utility of gain-of-function research is complicated. There's not a one-size-fits-all answer, I think. I think it's absolutely clear that certain types of gain-of-function research are really important and necessary to advance preparedness. But I also think it's true that not all gain-of-function experiments justify the risk, and that in many cases, there are ways to de-risk gain-of-function experiments. So maybe I could provide an example. It might be really important to understand how a pathogen might become resistant to an antiviral, right? If we know the mutations that lead to antiviral resistance, then we can survey for those mutations in nature. However, if you have a really dangerous pathogen, you might not want to make that pathogen resistant. And so, given sort of modern genetic and recombinant approaches, you might be able to do that same experiment and get that same answer by, putting the target of interest in a less pathogenic strain, right? So, maybe you're interested in an antiviral that targets coronaviruses, maybe it targets MERS, but you know that you can put that MERS protein in a seasonal coronavirus, not change the inherent safety, de-risk it. So it's all of that is to say it's very complicated, and it's very difficult to develop frameworks because of differences jurisdictionally across the world, right? So to come up with a framework that is shared by everybody is politically very complicated. But I do think that we do need to be very thoughtful about gain of function experiments. And I think as a overarching imperative, we should always be thinking about ways of de-risking those types of experiments so that we can get the critical answers we're looking for without creating disproportionate risk.
All right. Thank you, Dr. Miller. Dr. Smaill, you'd like to go next? Or...
I think the only input I would have is that as the medical monitor for the level 3 laboratory, I would be looking to ensure that whatever is done around some of these modifications is done in an environment with the training and the safety precautions in place. So I have nothing else to add except to ensure that these things are done in the right environment.
And as someone who works in the level 3 lab, I'm thankful for your constant medical oversight and the thought you put towards protecting us through that way. Dr. Banerjee, is there anything you'd like to add? Or?
Thank you, Michael. I agree with my colleagues. We work both with SARS-2 and MERS-CoV as well. And I can just speak for myself. We have extremely strict policies from what safety protection you'd wear. They're all separate and kept separate for both pathogens. So not just intentional gain of function, we're also very cognizant, and I'm sure my colleagues are also very cognizant of pathogens that shouldn't be located at the same place at the same time. So we are fully aware of those implications as well. So there are lots of checks in place. There are institutional oversight committees, there are national oversight committees to essentially avoid what's been suggested by this individual in the question. So I'm hoping that our best practices will avoid that from happening.
Thank you. And Dr. Poljak?
So I'm really not a lab scientist, so I don't have much really to add to this discussion, but maybe I would take a little bit of a different angle to this and say, we have a lot of genetic information, and there is always a desire to link genetics with some sort of phenotype. And I think with this field data that are collected, it will be really good to have standardized approaches to recording the phenotype data, because then we can not only have the experiments, the lab experiments, but we can also have additional data from field that will tell us something about the pathogenicity of the virus in one or more species, transmissibility, the host distribution, and things like that.
Excellent point. Okay. Thank you for that. On to our next question, which is that pandemics are by definition a global issue and therefore would require an international approach with partnerships around the world, both academic and otherwise. What is needed to help forge these international partnerships, and how could the Nexus help us bridge any of these gaps that currently exist? Would anyone like to go first? Dr. Miller?
Sure. Well, I think I think I'll come back to a point Dr. Smaill made earlier, which is, trust is incredibly important. And I think we saw some really serious mistakes during this current pandemic that eroded trust in certain situations. I think one of the examples that comes to the forefront of my mind are travel restrictions imposed upon South Africa after proactively reporting, novel variants, which I think were really unfortunate and didn't do much good, first of all, and second of all, disincentivize being proactive about reporting. I also think, we experienced issues of sort of, ethnophobia as a result of, this virus originating in China. That obviously was not helped by the US president at the time that the pandemic started and his choice of how he characterized it. But I think we just need to be cognizant of the fact that pandemics can start anywhere, right? The thing that I think is unique about Nexus is that one of the things that precluded effective responses during this pandemic is that all the critical players largely were operating in independent silos. And I don't mean so much disciplinary silos as I do mean sectoral silos. So public health was very siloed from academia, right? Which was very siloed from government, which is very siloed from industry. And all of these players need to have relationships and be talking to each other because they all have roles to play in both preventing and responding to pandemics, right? Our greatest discoveries don't reach the public unless there's someone to commercialize or implement them. As Dr. Smaill noted earlier, we need, our policymakers to have experts with whom there are long-standing trusted relationships to help to communicate the science so that policy can be implemented. And I think something else that occurred to me sort of after an earlier discussion that I think is something that's also very complicated in the realm of science communication is the fact that the science and policy are related, but they're also distinct from one another, right? Policy takes into account all kinds of things that don't necessarily marry perfectly with science, right? It has to consider feasibility and implementation and all kinds of things where, the science still hopefully is the driver, but there are a huge number of examples where the policy and the science aren't always married. And there's sometimes very good reason for that. And I'll admit that prior to, being involved to a certain degree in policy during the current pandemic, that's something I did not appreciate with the degree of sort of nuance that I appreciate now. And so this is where I think, Nexus can play a really important role is by, serving as a nucleus for people across sectors. I talked about government, industry, public health, academia, but also, I would say maybe with the exception of places like Guelph, where this is probably much more ingrained, human health and animal health people are not as integrated as they probably should be. And obviously there's been a tremendous push towards more sort of One Health type frameworks to address challenges associated with infectious disease. And I think that's really positive.
All right. Thank you, Dr. Miller. Matt just mentioned the connection there between human health and animal health. So I guess that's a segue to go directly to you, Dr. Poljak, for any additional comments.
Sure, I would be glad to add a couple of other things, and I look at that primarily from education and research standpoint. How can we increase international collaboration? And you know, in Canada is very international. The rest of the world may or may not be such international and intercultural society. And I think we need to understand also how the other countries and other cultures approach certain problems. And technology for that is available right now. I'm sure that policymakers, they travel and they interact with these people, but I think we have opportunities for our students also and researchers to collaborate with others. So I would emphasize really this international collaboration in research and international collaboration in education as well. And I think two avenues for that are probably One Health and global health as well. Because in my mind, they are kind of really linked. You know, in global health, you have to look at international collaboration, and the people are coming from different jurisdictions. In a way, you have to do exactly the same thing in One Health. People need to talk to each other, and maybe, you know, their education was initially siloed, but they need to develop this intersectoral and interdisciplinary approach in order to solve these complex problems.
All right, thank you. And I believe that you bringing up One Health there is now the perfect segue to pass this off to Dr. Banerjee.
Thank you, Michael, and I fully agree with my colleagues. The pandemics, by definition, are complex problems. And these problems require inter and multidisciplinary collaborations. I don't necessarily think we need to remove silos, but perhaps we need to align silos because we need subject matter expertise still. And that is something that One Health aims to do. Like, you take environmental health, human health, animal health, and you understand and appreciate the interconnectedness of all three sectors of health. And Guelph is a world-class institution that's leading One Health in our country. So I'm hoping there'll be more interest in funding because research needs money. And change is driven through science and research, right? In our country, historically, One Health has fallen through the cracks. You know, you would split your science to fit your science mandates with NSERC or CIHR or SHRC, but you wouldn't have a program that you could build a team to holistically drive policy changes at the level of a population driven through lab science and field science, animal health and human health. Where would you go for funding? But we're hoping that this will change because now we have a deeper appreciation of One Health in Canada. And hopefully there'll be more mechanisms to bring our colleagues together and do real One Health science.
All right, thank you, Dr. Banerjee. Dr. Smaill?
I'm going to take a slightly different tack at the moment because the effect of a pandemic on the health of the population is profound. I read in the Globe and Mail a couple of days ago that 2 children had died in Saskatchewan of tuberculosis. And so the effects of the pandemic are not just on the narrow focus of the infection with the pathogen that we're talking about. And I think we saw that very clearly with COVID. So the surveillance for other vital infectious disease pathogens such as tuberculosis, HIV testing and drug provision in Africa just about disappeared during the pandemic. And we also saw the morbidity and mortality associated with other infections and other conditions as a consequence of not being able to provide resources for the elderly. So what I'm hoping that Nexus will do will take that sort of global literally, but also holistic approach to the health of the community when a pandemic occurs, because I think if we narrow our focus too much, then we will miss an opportunity to actually help. And so one of my sort of hopes for this type of approach to pandemic is that we don't just narrowly focus on what's going on with the pathogen that we've identified, but we recognize how that affects the health of the community.
Yep, I think you're exactly right. And we actually had a guest seminar earlier this week from Dr. Pai at McGill, who gave a fantastic talk on the epidemiology of TB and its, treatment and lack of advances in both treatments for therapeutics as well as vaccine designs. And that has a particularly large impact here in Canada, up in the Inuit and Aboriginal populations, which are much more marginalized compared to places in big city centers. And I guess last but not least, sort of following up on something else that Dr. Smaill mentioned there in terms of the pandemic, not just having a direct effect in terms of COVID morbidity and mortality, but also at stressing some of the inequities across the globe, as well as here in Canada itself. And it also kind of curtails back to the issue of trust. Did the COVID-19 pandemic actually expedite our preparations for the next pandemic regarding things such as therapeutics or development of vaccines and formulation of policy? Or is it possible that at this point, then the politicization of everything and burnout, everyone's so jaded with some of these mitigation factors like masking and social distancing, that it might actually hinder the public health response in case of the next viral outbreak? Dr. Smaill, if you want to get us started, maybe.
I think in my 40-year career, I've never seen really such enthusiasm for science and for infectious diseases. So I have to think that if we don't take the positives out of what has really been a devastating pandemic for so many people, so many countries, we're going to miss an opportunity. We've recognized the need to really have infrastructure in our research laboratories, have communication, have open dialogue, have a multidisciplinary approach. We've also really come to appreciate how important people are. We have, we thought that if we just had a vaccine, everybody would take it. No, it's not like this. So a lot of those very important lessons have to be learned. And if we blow this opportunity, then we've blown it. But I think we've got something that we can now build on.
Thank you for that, Dr. Smaill. Dr. Miller?
Yeah, I mean, I certainly agree that, as a human species, we innovate in the face of, tremendous strain almost always, right? It's disaster that leads to innovation. We see these huge spikes in technological advancement in times of war, in times of environmental devastation, and we've certainly seen that in the context of this pandemic too. I am also, though, equally pessimistic about our propensity to only respond during times of disaster, and the extent to which we will learn the lesson of the imperative of being proactive in the wake of this pandemic. I think we forget that we're not new to pandemics, right? We've had 5 flu pandemics since 1918. We've had other massive global infectious disease outbreaks like the HIV pandemic, but also, we've had Zika, we've had West Nile. And I think there are lots of good examples, as Dr. Smaill noted, of lasting impacts. I think, the HIV pandemic, I would say, in my view, has had the most lasting transformative impact on the way that we approach infectious disease, not only as a result of the biomedical innovation that happened, but also because obviously HIV, especially in the early days, disproportionately impacted a marginalized population. And the way that we approach the human component of battling infectious diseases, I think was irreversibly eventually changed for the better as a result of what was learned in the context of the AIDS pandemic caused by HIV. Yeah, and you know, perhaps my pessimism is somewhat catalyzed by the relatively depressing recent provincial and federal budgets in terms of their stark lack of focus on science. But I think the idea that a penny of prevention is worth at least $1, if not $100 in response, is a really important message that we need to make sure that we convey. And I think that we as a scientific community in Canada need to do a much better job collectively lobbying government to underscore the importance of science. It frustrates me in Canada when we have such problems emphasizing the importance of science funding, and then we look to our neighbors at the south who are more polarized than they have ever been in their history. And the one thing that causes unanimous alignment of support is the NIH budget. Even Donald Trump authorized massive increases to the NIH budget. So I think that we, Canadians like to work together, and I think we just need to use our collective voice to stress the importance of being much more proactive in the way that we approach infectious disease challenges, and more broadly than that, the way that science can help us to avoid some of the other tremendous challenges that we face, including things like climate change and the energy crisis.
And just something you said there, Matt, kind of resonated with me, particularly relating this all back to the initial outbreaks of HIV and AIDS, 40, 50 years ago now. But we did, back then there was a lot of stigmatizing language, and, the gay population was, disproportionately blamed for the spreading of this pandemic. And, we would hope to think that we would have learned from that and all the trauma that we put some of these populations through. But I think we again saw some of the same messaging happening with the Mpox outbreak that was happening last year. So that's, once again, we have to sort of be humble and remember that, we're not perfect, and there's always lessons to be learned looking back in the past there.
I think Mpox, there's a very fine line, right? Because what happened with Mpox, I think there was almost an overreaction. I think we do have to recognize that specific interventions are really important. And I think, to Dr. Smaill's point, the fact that there had been a relationship of trust established between the medical community and the homosexual population actually facilitated very effective non-medical or non-pharmaceutical measures that led to mitigating Mpox. But you're absolutely right, Mike, that there was also the flip side of that, which was, stigmatization that led to the overreaction in the other direction. So I think approaching these things appropriately is critically important.
All right, thank you. Dr. Poljak.
I mean, these are all wonderful thoughts by Dr. Miller, and everything that I would add to this would be pale in comparison. One thing that I would perhaps add is that some of the consequences of the current pandemic are seen right now, and others are going to be seen in the years and decades to come. And one big thing for me is the supply chains and how we organize supply chains, because that has huge implications both on the global scale and maybe on the continental scale as well. So I think as a part of pandemic preparedness, we should probably consider some of this type of risk assessment as well, if things go astray, what can go wrong, and how can we mitigate that before that happens?
It's always important to have a plan in place and not be reactive, but be proactive to these sort of things. So there's no time to waste in the case of an eventual viral outbreak. And lastly, Dr. Banerjee.
Thanks, Mike. I want to be optimistic. I think we are trying to make progress in Canada, although our contributions or our budget's minuscule compared to our neighbors in the South. But we are trying to make that progress. I am worried, though, about sustainable funding. We're funding a lot of infrastructure in our country. We're funding a lot of biomanufacturing hubs. In fact, one of them is being co-led by my colleague, Dr. Miller. But I'm very worried about how are we going to sustain this long term? Like you said, wartime money flows in, but peacetime, how are we going to keep all of these infrastructure up and running? As you're well aware yourself, Michael, training in CL3 or in high containment labs is no joke. And you have to maintain your training, right? You can't be like, oh, peacetime, I'm going to go do something else. There's a pathogen and I'll jump right in. That's not how it works. So you have to be trained. You have to be up to speed with your training. And you need to be paid to stay trained during peacetime. So I'm worried about how that sustainability is going to show up in our laboratories.
All right. Thank you, Dr. Banerjee. And I don't think anyone has any closing remarks. We've answered all of the questions in the chat. So with that, I'd just like to thank all the panelists for taking the time out of their very busy schedules to have this conversation with us and for sharing with us some fantastic insight that can only be gained after years of study and passionate research. And I would also like to thank everybody in attendance for their participation in this discussion. So please stay tuned for the next edition of the Global Nexus Conversations. And I hope that everyone can now go out and enjoy the nice weather and have a wonderful weekend. Thanks for doing such a great job moderating, Mike. Thank you very much. All right. Goodbye, everybody.
Thanks, everybody. Bye.
Okay, goodbye.